Hello, everyone. My name is Andrew Eriksen. I'm a director on the advisory team here at Allele Capital. I want to welcome everyone to today's webinar titled Compassion in Action, where we will be hearing insights from a compassionate use study that observed 46 late-stage cancer patients and their compelling responses to a proenzyme formulation. What we are looking to accomplish today is to provide a comprehensive background on what exactly was this compassionate use study, what was observed, why it was so compelling, and how that eventually led to the creation of Propanc Biopharma, and what Propanc is doing today to push this proenzyme formulation forward to help those battling these deadly cancers. Before we kick off the call today, I want to briefly introduce all the speakers on the call, all of which who are experts in their respective fields and who are also part of the Propanc management team. James Nathaniels is our first speaker, and he is the CEO, CFO, and executive chairman of Propanc. He has served in these positions since October 2007, and prior to Mr. Nathaniels serving as the CEO, he was the new products manager of Biota Holdings Limited, an anti-infective drug development company in Australia. Mr. Nathaniels is one of the co-founders of Propanc Biopharma and has extensive experience in R&D, manufacturing, and distribution. Next, we'll be hearing from Dr. Julian Kenyon, Chief Scientific Officer and another co-founder. Julian co-founded the company and was appointed as a director in February 2008. Since 2000, Dr. Kenyon has served as an integrated medical physician and medical director of the Dove Clinic for Integrated Medicine in Winchester and London. He was the founder and chairman of the British Medical Acupuncture Society and co-founded the Center for the Study of Complementary Medicine in Southampton and London. Propanc is based on Dr. Kenyon's initial work at the Dove Clinic, and he is the one who ran this compassionate use study that we will be highlighting today. So I think I speak for everyone when I say we are extremely excited to hear his insights on the study. Next, we'll be hearing from Professor Klaus Kutz, Acting Chief Medical Officer, who has more than 25 years of experience as an independent consultant in clinical pharmacology and safety for pharmaceutical companies and clinical research organizations. Professor Kutz's specialty over the last 6 years has been in oncology, including preparation of multiple NDAs and INDs for small and medium-sized pharmaceutical companies. Dr. Kutz is also professor of medicine at the University of Bonn in Germany. And last but certainly not least is Dr. Ralph Brandt, Scientific Advisory Board member and co-inventor of Propanc. Dr. Brandt is a biochemist and cell biologist, also with over 25 years of experience. His expertise is in research programs of experimental oncology, and he is also the co-founder of VivoPharm. Since 1990, Dr. Brandt has been active in the field of preclinical oncology as he led the tumor biology program at Novartis Pharma. And during his long career in the pharmaceutical industry, he has acquired significant knowledge and expertise in leading business units and representation of services to the preclinical research market. I think it's clear that all of our speakers today have extensive experience and knowledge in their respective fields, and as such, I'm excited for them to share their perspectives and opinions with everyone on today's call. So with that, let's dive right into the discussion. I'd like to start with setting the stage with the global cancer market and highlighting its importance and relevance in this context. I think most people listening understand that cancer is a large, growing market, which is estimated to be roughly a $250 billion market by 2027, with the metastatic market contributing to nearly 50% of that. In connection with this massive clinical and commercial need, it is well known that there's a large amount of players in this field going after what seems to be an increasing amount of various cancer types. Keeping this all in mind, I want to open up today's discussion to the panelists and ask, why do you think we need another cancer treatment in today's market? Maybe you can provide some insights to the market as a whole and highlight any deficiencies you have seen in your personal and professional experiences. Maybe we can start with Propanc CEO James Nathaniels and have some of the panelists chime in with their additional thoughts. James?
Well, thank you very much, Andrew. The first question that any patient asks when a tumor is identified is whether it's malignant, whether it's spreading. Now, traditionally, companies have been very good with established forms of treatment at attacking the primary tumors. But the fundamental question remains, which is, did we get all of it? Is there a chance that cancer will return? And that's largely due to metastatic cancer, which is as a result of tumor recurrence. What we are developing is a new novel technology that can stop the spread and return from cancer, but without the severe and serious side effects from treatment. And this, we believe, is still very much a fundamental gap in the market.
Great. Thank you for that, James. Is Professor Kutz, Dr. Kenyon, Dr. Ralph Brandt, do any of you want to add to that, or did James basically knock that off?
No, I think it is important.
I think that's enough, basically.
People, sorry, Julian, please continue. You wanted to see something? No,
go ahead, Professor Kutz.
Oh, no, it's okay. Thank you. I think what is important is what James spelled out. First, we have successful treatments for the primary tumor. But then when we are going, when the tumor, or at the moment when we are making surgery, we do not know at this moment whether the tumor has already spread. And therefore, I think we do a lot of investigation thereafter to look whether there are metastases. And the problem is then after surgery, the second approach is chemotherapy today. And chemotherapy, as Julian says, is very toxic, and it has a lot of adverse events, sometimes very fatal adverse events. And therefore, an approach to treat tumors without these serious adverse events which can occur, this would be a big advantage. Julian, I think you agree.
Great. No, that was extremely helpful. Thank you, Professor Kutz, and thank you, James, for your initial commentary. I think that sets the stage nicely as to why there is room for another therapeutic option, and there are still major gaps that exist in the current treatment landscape, and that's something that the Propanc team is looking to address. So, with that background set, I think let's just dive right into this compassionate use data that we've been referencing, because it is truly inspiring in the core of what we want to cover today. So. Dr. Kenyon, I think I would direct this to you. Given the fact that you ran this study out of your lab, I think it's best to start with your insights. And can you please provide the audience with some, background on what this study was, how and why you decided to run this compassionate use study, and what you initially observed?
Yeah, cut a long story short, I've published widely in the peer-reviewed medical literature. A significant number of these papers have come from Propanc. I've been treating, by the way, my papers have received over 700 citations over the last 5 to 6 years, and that's absolutely outstanding. There's a lot of interest in what I'm doing. I've been treating cancer for over 40 years now. The compassionate use of the pancreatic proenzymes and amylase, these were no-hope patients. They'd failed everything. They'd failed all conventional treatments. They'd failed also a range of other treatments I regularly use for cancer. By the way, at the moment, I've retired from clinical practice and I'm devoting entirely all my energies to Propanc because I think that's the most important thing to do. Now, all these patients were complete failures. I did not know what to do with them. So I decided to use compassionate use of the pancreatic proenzymes. I didn't expect to get any results at all. That's the first thing. The second thing is I expected to get some side effects, but we had no significant side effects. Now, that's truly extraordinary in cancer treatment. The majority of cancer treatments have significant side effects. Some of them are lethal. Then we started to get these most extraordinary results with pancreatic cancer, for example, dropping of tumor markers. I was just completely blown away with that. In ovarian cancer, with lowering of tumor markers. With prostate cancers, all the markers, PSA and so on, remain stable throughout treatment. And I suddenly thought to myself, this is just totally unexpected and outstanding and without side effects. And what went through my mind is that we had something truly extraordinary here, which was safe and there was a good background bunch of research to support it. And then then I met James and so on and so forth. But by the way, my patient population came from all over the world, the States, from Africa, from Europe, from Australia, and so on. So I'm very much internationally sort of focused, really. So I thought to myself, this is just truly extraordinary. And I met a friend of James and myself, and we decided to start Propanc because we got something truly unique, and also the whole mechanism was completely innovative and new. Nobody's ever tried this before. Absolutely nobody. And it's also relatively cheap. Now, that's massively important because cancer treatments cost a lot of money. And it's very likely that once we get this off the ground, clinical trials and so on and so forth, that there will be a lot of official interest from health HMOs and all the rest of it in using this kind of treatment because it doesn't cost so much. You can do more patients.
Yeah.
So do you want to ask me anything more on the basis of what I just said, or?
Yeah, absolutely. No, I think that was a great background. I appreciate that insight. And I think one of the comments that you had made was, no one had really tried this before. Your patient population was widespread, coming from various countries. And my question to you would be: was there any insights or data that suggest that all these patients had in common, besides the fact that prior lines of therapy weren't working for them, that you would attribute to the success of this treatment, or any other attributes that you'd want to flag as it pertains to these particular patients?
No, they were all varied cancers. And that's what's extraordinary, because most cancer treatments work best on particular sorts of cancers. Right. And so the thing which simply blew me away was, look, this was working on all the cancers we treated, and there was a wide range of cancers in the cases we looked at. So this was completely, totally unexpected, cheap, effective, and no side effects. So I thought to myself, I've got something extraordinary here.
Yeah, no, that's incredible, and I think everyone agrees here that it's, especially when you're dealing with patients with limited hope based on the prior lines of therapy, and you see such a positive outcome, it's extremely rewarding, I'm sure, and obviously got you excited about what this could be potentially. So no, I really appreciate that background, Dr. Kenyon. And go ahead.
You're going to add one more thing. These were end-stage cases. I didn't expect to cure anybody. I didn't cure, and in end-stage cases, you don't cure, that basically, I wasn't curing them, but they were staying alive and lowering your tubular markers and all the rest of it. So I thought I'd got something extraordinary here.
Right. And I think we can all agree, when you are in that stage, even the extension of life by a few months is remarkable, and something that people are looking for, even when it might be a more sorrowful outlook there. So no, that's great background, and really appreciate that, Dr. Kenyon. Super insightful, super helpful. I'd like to actually jump over to you, Professor Coutts, because I believe you put together a retrospective analysis of this data that Dr. Kenyon did, and so I'd like to hear a little more about what did you take away from that?
In my medical history, I have seen a lot of different cancer patients with very limited life expectation. And I was surprised when I saw the first data that Julian has, from patients that Julian has treated. In total, there were 47 patients, and with really advanced malignant diseases. And there were some patients with prostate cancer, there were patients with GI cancers, some with breast cancers, pancreatic cancers, ovarian cancers, etc. And the life expectations varied, depending on the carcinoma, varied between 2 months and 12 months, a year or so. And when I look to the survival, the patient who has a 2 months life expectation based from literature data, it survived 8 months. The same later on for pancreas carcinoma, instead of 3 to 4 months, they survived 7 months or years, etc. So there were a really impressive data coming from this raw data that Julian has created, and this was then later on the point when I said to James, this is a very interesting project. We know that Julian has given the mixture as suppository, but I think I know from my pharmacy background that suppository sometimes have not the best bioavailability, in particular when we are speaking from proteins. And therefore, I think this was then the switch when when James gave the compound to Ralph to do toxicology in a parenteral route of administration, no suppository. And so we are standing here now with a formulation that is easy also to apply. And then I think we have done then a tox program that allows us then go to human in the first clinical trial in patients.
Right, of course. No, very helpful. Thank you, Professor Kutz. And just for the audience too, in case you didn't hear it, the initial formulation that was used was this 1-to-1 ratio of trypsinogen and chymotrypsinogen, as Professor Kutz just alluded to, which was administered through a suppository, and that's what produced this exciting clinical results. And as we'll get into in a second here, Propanc has taken that formulation and enhanced its synergistic ratio to hopefully see better results in this trial that we're alluding to. But thank you both, Professor Kutz and Dr. Kenyon, for your detailed background on this compassionate use study. I think that helps provide the audience with some background on what the data showed us and why it's so powerful, especially when you take into consideration, as Dr. Kenyon alluded to, that many of these patients had prior lines of treatment with no success. There's really no optimistic outlook for them. And even extending a patient's life for a few months makes a tremendous impact on the patient's life. And I think that's what's really exciting with what the Propanc team's doing here. So now that we've provided this background on what this compelling compassionate use data is, I want to transition to Propanc Biopharma as a company. James, let's pass it back over to you, because maybe you can provide the audience with the background on the company. And I think, something that we just talked about, one of the more interesting parts of the Propanc story is the creation of the company off of this compelling data versus, a company already existing, running some early trials and using a compassionate use study to rationalize what, you're working on in those early trials. So maybe start with there and, provide the listeners with an overview of what Propanc is focused on today.
Right. So as mentioned, that compassionate use study, we had it independently reviewed at the time through Professor Kutz's work. And collaborating together in a roundtable discussion, Dr. Kenyon and I felt very strongly about the potential of this technology and what it could potentially do longer term for patients if we can catch them early enough and treat them for long enough. So we went through a formal, rational pharmaceutical drug development program. Very early on in the piece, when we formed the private company here in Melbourne, Australia, we held scientific advice meetings with the MHRA, with the UK health authorities, and we predetermined what we would need to do to take this drug product forward. And so we started to develop a non-clinical program through the expertise of Dr. Brandt, and we advanced and identified a formulation. Now, what's important is that these proenzymes are naturally derived. And so what we had to do was to discover and understand these proenzymes so we could build and develop our intellectual property portfolio and become more investor ready. And we managed to do that. We've raised significant capital, primarily through US investors and becoming public in the US. We've undertaken our non-clinical program with the oversight and guidance of Professor Kutz in collaboration with Dr. Brandt. And as Dr. Kenyon mentioned, we identified research partnerships, which has helped us to elucidate the mechanism of action, characterize this drug formulation, which is now ready to advance from a regulatory standpoint into clinical trials. So we're very excited about this tipping point, but it took some time to get there. And I think Dr. Brandt would be able to share his insight because he was a co-inventor on what we subsequently discovered would be our foundation patent family that's filed around the world.
Yeah, that would be that's super helpful, and I think that'd be really insightful from Dr. Brent as well. So maybe Ralph, you can talk us through that optimization of that formula and some of these initial patents that James just alluded to.
Sure, certainly. Maybe I want to say first that when we founded VivoPharm, we had the ambition to have a CRO, so a preclinical testing research company with a different profile, to have models in hand and procedures in hand which are more predictive, hopefully, for the clinical outcome, even using preclinical models. So which is one of the missing parts from a lot of models to get the results forecasted for the outcome in the clinic. So VivoPharm was specializing at that time when I met with James in two major parts which were driving our relationship for success of that formulation. That's combination studies in vitro, means in a petri dish, and also in vivo with animal models. And the other part is orthotopic tumor models, whereas most of our competitor CROs had injected anti-cancer drug candidates into animals with subcutaneous tumors. But it is absolutely clear that tumors are not growing under the skin somewhere, so they are in the target organs. And we have specialized in these target organ models called orthotopic models and have good models for pancreatic cancer, colon cancer, and ovary. And so that maybe as an introduction. When I got contacted by James with that original formulation, I immediately asked the question, is the ratio of the two components, the proenzymes, optimized? Has anybody looked into it, whether the one or the other is acting better in a higher or lower concentration? So that we did then in vitro studies against target cell lines for the cancers mentioned, to see whether there is any optimization possible about the ratio. And we came up with a ratio of 1:6, or 6:1 the other way. And that has provided us much better outcome already in the petri dish, which has then been translated into our animal studies with pancreatic, colon, and ovarian cancers, with a very, to me, surprising outcome as well, as Julian mentioned, because there was no historical data. And normally people would say that it can't work because it was not clear what the mechanism is, which had been then elucidated a little bit later, at least from a point of a hypothesis, backed up with some data. So we got impressive results in inhibiting tumors up to 70%. I have to say here also that our setups of models were going into the direction only to inhibit tumors. So we had not looked into questions like differentiation of cells or any change in morphology of cells. And knowing that in orthotopic tumors, you have a lot of interaction with the stromal cells surrounding the tumor, we had an idea that there is a special effect on changing that interaction, but we didn't study it. So that was then later done by partners with Propanc, which have then found that the interaction between the stroma and the tumor cells, or the tumor itself, is changing. So that process was not called epithelial mesenchymal transition. And that is also inhibited by the formulation, which was found later. And that is an explanation that in principle, maybe we don't see a total regression of tumor. We see a slowdown of tumors because the tumor cells are reversing into their epithelial state. meaning they are more differentiated. So, and we know from outcome of clinical studies, I hope I say the right, Julian, that more differentiated tumors have normally a better outcome. So if we can reverse the effect of these tumors, at least here also in the animal model, to bring them back in a more differentiated stage, then there is a very good chance that you have a longer disease-free outcome. And maybe combining, and that's an idea I would bring in further discussions of models, in combining it with other types or metronomic therapies, you might have a very long-lasting survival effect with that treatment. So taking that all together, we had a surprising outcome, as Julian had, because that was interesting. But we also believe that we got these good data and the explanation because we used the right model. And that was a discussion I had with James and Propanc, because you would not see that in a subcutaneous classical tumor model. With the autotopic model, you have a more natural occurring blood vessel system, so that the angiogenesis, so the building of the blood vessels, is more natural, and therefore, and we have shown that for other larger molecules, the distribution and delivery of the drugs is much better than you would never see it in subcutaneous models where you have a totally artificial system. So therefore, I'm looking forward to maybe additional things in that respect, especially also clinical study. And I want to finish my presentation with some words about toxicity. So there was a lot of discussion about less side effects, and I think it's a major point. If you have a chemotherapy for a patient, you normally start doing that calculation, how many months you have a low level of quality of life because of the chemotherapy and, feeling miserable, and then the extent of the survival after the therapy. So we don't see any of these effects with the Propanc formulation. So we did toxicology study in a setup which you would do for a normally IND application. It was a repeated dose 28-day tox study in rats. And we could demonstrate that there's a dose-dependent light effects on blood cells, and maybe also that you have a load of protein, but nothing severe to indicate a toxicology in any outcome we see with the chemotherapy when we do such studies. And also the dose range was clearly indicating that the dose used for the tumor treatment is safe. So taking that together, I'm really looking forward with all that set of data, which is available now, to move it into an IND application. I believe it's not too much to do anymore on the preclinical front here. And it will be a very quick transition into clinical studies with predicted good outcome. And coming back to the market question, the market is the one thing from an economical point, and that's what you're representing. But I'm looking also market in relation to patient. Yeah. Because I still remember that Novartis story about Gleevec. Yeah, when people learn there's a good drug out there, the patient actually were driving the development of the drug. And I have the same hope here with that formulation. If there is clinical data coming out and the survival is shown, that the patient will be very interested, and that's then of course driving the market and the financial situation.
I think this is something which sometimes is. I have frequently observed also with orphan drug or orphan indications that the patients later on are driving the development and forcing the development, which is then sometimes comes to a very positive end.
Absolutely. No, having that patient-centric perspective is, I think everyone can agree here that's first and foremost, and that is what feeds the commercial side of it, of course. But obviously, improving patients' lives is the number one objective. So,
and maybe
go ahead, Dr. Brandt.
Yeah, also adding pancreatic cancer and ovarian cancer. These are two cancer types which are hardly treatable. So that the median survival time on these cancers is less than 3 years, I think, if I'm correct.
Yes.
And sometimes even faster. I've personally had experience now with pancreatic cancer in the family, so it's very rapid. And therefore, I think these targets and having these good outcomes, again, will be patient-driven if that is going into public knowledge.
Yes, and therefore, I think what's also very important later on when we are starting clinical development is the quality of life for patients, which should be completely different to the quality of life if you have a cancer, cytostatic or cytotoxic treatment at the end. And I think the quality, and this I've seen in some clinical trials in patients, this is very important for the acceptance of the treatment.
Absolutely, absolutely. Because if it's not improving your quality of life, if there isn't a high level of safety profile, then some patients might not even bother wanting to try to take it because it makes their quality of life that much worse. So that safety profile is such an important thing here, and I think it's a very strong message that's clearly a focus for Propanc. And so just coming back to this discussion here too, is, essentially you came across this compelling compassionate use data, which reported outstanding results both on the efficacy and the safety side. You took that formulation and optimized it so you could still have that superior safety profile, but hopefully we're going to see some improved efficacy with these future trials. And James, you alluded a little bit to an upcoming clinical trial. I think it was a phase 1b study that you might have mentioned. You mind providing the audience with some more details around that and what that's going to look like and what the objectives of that will be?
Sure, no problem. I can give an initial high-level summary, and then I would defer to my Chief Medical Officer, Professor Kutz, to perhaps provide some further insight. But what we discussed as a development team with management, as well as some regulatory consultants that Professor Kutz introduced and we worked collaboratively with, we determined that we would do a phase 1b study in 35 to 40 patients approximately, suffering from solid tumors. So what they call an all-comers study. Now, this would be a classic maximum tolerated dose study with a few cohorts, carefully increasing the dose in a stepwise fashion with a minimal survival or life expectancy at around 3 months. What we're looking for is to primarily identify that maximum tolerated dose so that we could go into a more detailed analysis in phase 2 in pancreatic and ovarian cancers. Now, those indications were specifically chosen based on the insight of all three of my colleagues here today, Professor Kutz, Dr. Kenyon, and Dr. Brandt.
Amazing. Professor Kutz, would you like to add to that as well?
No, I think James has already laid out the procedures of our clinical development. And I think we will start really with a classical phase 1 or tolerability study in cancer patients, which is limited to a small group of end-stage patients. And so it will be a stepwise increase of the doses until we have reached our maximum tolerated dose. We start with one patient, and when this patient has the dose well tolerated, we are increasing the number to 3 patients, and then look what has happened. If we have some adverse events, which we think are serious or severe, then we enlarge the group. So at the end, we expect that we at about 30 patients, perhaps 2 or 3 patients more or less, then we have reached the maximum tolerated dose with which we can go later on in our real phase 2 project. That means our phase 2 study in patients and we have selected pancreas cancer, and ovarian cancer, but also could be discussed is prostate cancer. I think what is important is that we look in this study, in the very first study, that we are looking to safety tolerability globally, that we look to targets which are targets for up to now cancer drugs, cytostatic, cytotoxic drugs, and quality of life, and also the pharmacokinetics. Pharmacokinetics needs to investigate exposure of the drug in humans and perhaps get some ideas also on metabolism and also on disposal and routes of administration, whether these routes are renal or fecal, or whether it is completely metabolized in the body. What we need to know in this stage, whether there are some organs or some processes which are rate limiting, and this we get by pharmacokinetic investigations.
Interesting. Interesting. And James, I believe you said this phase 1B timing, that would be around 12-month trial. Is that right?
Sorry?
That's correct. So it would be expected to be a time duration of about 12 months.
Yes, of about 1 year. Yes, not longer.
Yes. And
gotcha. And sorry, I was just going to say that then you alluded to running, a phase 2 study in 2 different indications. Is that something that would be run at the same time, or would those stagger each other? How are you thinking about that, assuming positive results from the phase 1B?
My preference would be first as a phase 1B study in the tolerability study as a multi-center, perhaps multi-country study, because what we need is a fast recruitment of the patients and fast results. And if the results are positive, we can do, when we have the resources, we can do 2 studies in parallel. I think there is no negative thinking about this. If the first study is positive, is well tolerated, we know about disposal and of the metabolism or whatever is distribution, then there is no reason to say, no, you should not go in parallel with 2 studies, but this depends on the resources.
Yeah, and coming back to resources, there's also a longer term chronic toxicology study for longer treatment. So in between, there has to be done a chronic tox study, I think at least with 6 months.
Half a year.
And that would mean that there parallel of these studies most likely technically is not possible, because you would have to produce these toxicity study data first.
What we do not need is a cancer study, which takes 2 years. It is not necessary in this indication.
Wow, interesting. Yeah, no, that's really interesting, and I would say it sounds like there's a clear path forward for the company and the development of PRP, which, as we know, is the lead asset. And I'm sure patients battling these deadly cancers and their families are extremely happy to hear that. Not only is there a long-term, intermediate, and near-term plan, but it's being run in the most efficient way possible, which is obviously what people want to hear and see as well. So, that's really reassuring.
Could I add something to that?
Absolutely, Dr. Kenyon, go ahead.
I anticipate that we'll have a very good sense of what kind of results we're getting in much sooner than a year, maybe 6 months. And in the cancer field, there's no competition for us, because this is safe, cheap, and effective. This is going to be a blockbuster, without any question. It's going to end up being a blockbuster.
I think, Julian, sorry for saying something, some comment, what you have said, the velocity of development, this is generally not so fast as we would expect. And therefore, I think less than half a year for the very first study, or less than a year, is unlikely. And later on, I think depends on the progress of the study. We need then a DSMB in the meantime that follows the study, and we can make immediate interim evaluations after half of the patient foreseen, etc. So see whether it is safe and whether it is also shows activity or efficacy. Ralph?
Yeah, and maybe I want to interpret a little bit what Julian said. Is what we have seen with other blockbuster drugs is that in the phase 1 study setup, which is not the goal of the phase 1 study, is seen already a positive outcome. And if that would happen here, and that combined with a safe approach with no side effects, which we do not expect, because we have the the companion use study already and know that that is quite safe. And then if there's any data coming out, as we know from the previous studies, as a extension of or reduce of tumor growth, static tumor or good outcome, that would drive already results even before these studies have been finished. So that, I think that's the idea from the data we have at the moment, and the prediction of the first results seen even in the dose escalating MTD study.
No, I think
Right.
you are right. I think in the dose escalation study, we have always activity. We have surrogate markers that we can follow. And then I think at the end, we see whether a patient who has already received this compound survives longer than expected. Yeah, but I think, unfortunately, clinical development takes time, and we need to stay in very narrow contact with authorities. And we have to explain to authorities frequently, first, our strategy, second, our goals, and third, how we can reach goals. I think interaction with authorities is important in this field. And what we can get at the end, we can get a easier development. As they say, yes, you must not do this and this and this, but how shall I say it? It is that you can get permission to bring the compound on the market, but follow up with safety data when it is already on the market. Conditional approval, for example. So it's a technical term.
Can I just come back in there? The treating oncologists will soon get a sense of whether this is working, and they will push the timetable forwards. This is what's happened with checkpoint inhibitors, which are now commonly used all over the world. That took 4 to 6 months before that really got going. And this is what I think will happen with us. So I think this is going to be quite a sort of a very interesting ride for us, and we'll all be flat out. That's a good job I've retired from clinical medicine.
I might just summarize at this point. It's been a wonderful fascination to hear from each of the experts. So with our phase 1b study, it is primarily a maximum tolerated dose study, but we will be introducing some variables, secondary variables that we will measure around some efficacy endpoints. Simply put, the RECIST criteria, which is a standard method of measurement in clinical practice to assess the control of how we regulate those tumors and observe their characteristics and the effects on the patient, but also a combination of that and the immune-related response criteria to have a look at survival, look at some secondary efficacy endpoints during that phase 1b. But our primary goal will be to identify that maximum tolerated dose, understand the safety profile, so that we can move into a proper phase 2 study in 2 indications where we can really understand and maximize the effects in specific target patient populations.
It is important if you can do, for example, the phase 2 studies in parallel, indication 1, indication 2, to see whether this organ is more sensitive to the treatment or the other is it. And so that at the end we can focus on one indication first, bring it on the market, and then if we are successful, then the indication may be broadened. I know this from other compounds as well. And first safety, tolerability, and then a parallel phase 2, and then focus on the most promising indication.
Gotcha. Just to put a cap on this point specifically too. There was a comment earlier made about conversations with the regulators, and James, I think it's in the presentation that there is an orphan drug designation already for the two,
Yes.
right?
Yes.
Some
So
of them you may get it, yes.
yes. So we were fortunate enough that the clinical cases we saw in the compassionate use data, in particular pancreatic and ovarian cancers, which are our target indications in phase 2, we determined that pancreatic cancer in particular qualified for orphan drug designation.
Yeah.
So with our preclinical package that we carefully constructed over time with Vivo Farm, with Dr. Brandt, as well as the clinical cases from the compassionate use study, we were able to obtain orphan drug designation for pancreatic cancer from the Food and Drug Administration, the US FDA. So this is something that at preclinical stage of development is not the usual practice, but based on the compelling clinical cases supported by the non-clinical data, we were able to receive this important designation. And we look forward to assessing this in pancreatic cancer with the FDA support as part of the program.
Amazing. Well, I think that's a good point to kind of put a cap on this discussion. Want to leave a little room for a couple questions here, but I just want to say, thank you to everyone joining this call. And, we learned a lot from the compassionate use data, how compelling that was, why that was so compelling. And, to the point where, it formed Propanc Biopharma to go after these, deadly cancers and trying to cure for those. So before we wrap up here, we want to just take a few questions from the audience. So we'll give ourselves 30 seconds or so to collect some of those questions. But in the meantime, while we wait for those to come in, I just want to comment again how exciting it is to hear about all these updates and potential milestones coming up for Propanc and the team here. It seems, what I personally find to be truly fascinating is that the team is taking a novel approach to treat these deadly cancers, but there's an elevated sense almost that there's slightly more de-risked than your traditional biotech claiming to be novel, because you do have this human data in hand heading into your phase 1B trial. Do you think that's somewhat fair to say, James?
Oh, yes, most definitely. We have spent 15 years carefully constructing our programs to get to this stage of development. This wasn't just done by coincidence, but where our competitive advantage lies is the scientific body of evidence, not only compiled by Dr. Kenyon and my colleagues, but also over 100 years of supportive scientific evidence. But we're the first ever to take this drug, put it to pharmaceutical standard, and look to explore its utility in target cancer patient populations. And we think it can make a significant difference. And as you can hear from my colleagues, we have a strong belief in the potential of this drug.
Very good. So let me just pull up, looks like we got one question here, which, works out because due to time, I just want to make sure we're cognizant of everyone's time. So while we're here, we'll just ask this one. Okay. It says you claim your drug doesn't have many side effects while most other drugs targeting these types of cancers do. Can you explain further why you believe yours is safer? What can you contribute to being less toxic? And do you anticipate that safety profile even as you get into higher doses with your phase 1 trial?
Yeah. These pancreatic proenzymes are naturally occurring, and therefore they do not have the side effects that we get from chemotherapy, from immunotherapies, and so on and radiotherapy. That's why. Now that's fundamentally important, because that puts us in a completely different field. And look, if we reacted to pancreatic proenzymes in our bodies, we'd all be dead. And the answer is we're not. So it's as simple as that.
I think, yes, there is a clear difference. I think what Ralph has spelled out at the beginning, it seems that this combination modifies the cells, make it from aggressive cells to less aggressive cells, And this is one thing. So this is a mechanism, important mechanism. And second, there is no reason for cytotoxicity. I think we are not, we have no neutropenia in the blood. We have no cell damage in the GI tract, etc. So no hair loss. We do not expect these things. Therefore, I think the negative sides of cytotoxic or cytostatic compounds, we don't think that they occur here due to the mechanism of cell modulation and not cell toxic.
Maybe to add also the term therapeutic window. Yeah. So what we see from the tox data and also the efficacy tests we did with orthotopic tumor models, we see that as a large therapeutic window, which for chemotherapeutics, despite all the fact that the side effects are very severe, but the therapeutic window normally is relatively small. And here you get a larger window, which allows you, in combination with positive effects on the cells, pushing them into differentiation, to have a much better outcome and quality of life for the patient.
And I think what I would expect, if I looked again to the data from Julian in the end-stage patients, so I would anticipate that the exposures and the plasma concentrations that we need must not be too high, because it was the suppository that he has applied. And as I said at the beginning, suppositories have normally not the highest bioavailability. They have a quite low bioavailability with a large variability also.
Perfect.
I anticipate that the doses that we need must not be too high, which is in line with that what you said, Ralph, with regard to your therapeutic window.
Fantastic. No, I think that was a very detailed answer. I think that knocked out of the park with answering the question here. So appreciate everyone's participation there. I see we have a few more questions that came in the queue here, but we're just pressed for time here. So what we'll do is we'll be sure to follow up directly with everyone who submitted these questions that didn't get answered live here. And I just want to take another brief moment here and thank all of our speakers for participating today. I know we're juggling a few different time zones, from the US to the UK to Australia. So I think we got every time of the day on the video right now. But I want to thank you all for your flexibility with getting on the phone today and your expert insights on the call. And thank you to everyone who joined in to listen today. We hope we were able to provide you with some additional detail and background on what you may or may not have already known about Propanc Biopharma and their exciting compassionate use data. So we invite everyone to continue to follow along with the story, and you can do that by visiting propanc.com or visiting Propanc on any of their social media channels, such as LinkedIn and Facebook. And we thank you all again, and we look forward to keeping you up to date on the Propanc story. So thanks for joining.
Okay.